All right. The next part is regarding female reproductive structure. Right? Female reproductive structure. Female reproductive structure, gynoecium or pistil. Right? It represents this gynoecium or pistil. It represents the female reproductive part of the flower. If it consists of a single pistil or carpel, it is known as monocarpellary. Single pistil or carpel, monocarpellary. Or it has more than one pistil, it is known as multicarpellary. More than one pistil. When there is more than one carpel, they may be fused. Then the pistil is known as syncarpous. More than one, and may be fused. Then it is known as syncarpous, or may be free, known as apocarpus. Each carpel has three parts, namely stigma, style, and ovary. Stigma. It is a landing platform for pollen grains. Landing platform for pollen grains. Landing platform for pollen grains. Style. It is the elongated slender part beneath the stigma. Style, stigma. Stigma. All right, stigma. Ovary. It is the basal lower part of the carpel. Inside the ovary is an ovarian cavity known as locule. Ovarian cavity known as locule, where the placenta is located. Placenta contains ovules or microsporangia. Ovules are microsporangia. The number of ovules in an ovary may be one, as seen in wheat, paddy, mango, etc., or many, as in papaya, watermelon, orchids, etc. What is a megasporangium? See a view of typical ovule. Chalazel end, nucellus, embryo sac, outer integuments, inner integuments. This one is also nucellus, micropylar and micropyle. All right, micropyle. Here only it will connect. The pollen tube will connect. Then, regarding the chalazel end, there are three antipodals. Then polar nuclei, a central nucleus, central cell is made up of two polar nuclei, two multicell, and synergids. These are synergid and one egg cell. This is known as filiform apparatus at the micropylar end. This egg cell and synergids combined together, it is known as egg apparatus. It is known as egg apparatus. Megasporangial ovule. It is a small structure attached to placenta by a stalk called funicell. The junction where the body of the ovule and the funicell fuse is called as helium. Junction where ovule and funicell, body of ovule and funicell fuse is known as helium. Each ovule has one or two or sometimes three protective covering called integuments. Outer integument and inner integument. The integument encircle the ovule except at the tip where a small opening called micropyle is organized. Micropyle is organized. Micropyle. All right. The opposite to the micropyle end is the chalaza, which is the basal part of ovule. Basal part of ovule, chalaza. Within the integuments, there is a mass of cells called nucellus, which contain reserve food material. Inside the nucellus, there is an embryo sac, which is also called the female gametophyte. An ovule has a single embryo sac, usually formed from a single haploid megaspore. Single haploid megaspore. All right. From single haploid megaspore, there will be division. There will be division, and which informs 8 different cells and 3 would be here, 2 would be combining to form central cell, and automatically here 2 synergids and X cell, combining this total one. So what is megasporogenesis? The formation of haploid megaspore from diploid megaspore mother cell as the result of meiosis is called megasporogenesis. A single megaspore mother cell is differentiated in the micropylar region of the nucleus. The megaspore mother cell, megaspore mother cell, MMC, is a large cell containing a dense cytoplasm and a prominent nucleus. The megaspore mother cell undergoes meiotic division, resulting in production of 4 haploid megaspores. Meiotic division to undergo 4 haploid megaspores. In most of the flowering plants, only 1 megaspore is functional, while other 3 degenerate. Out of 4, 1 will be functional and other 3 will be degenerating, giving nutrition to the 1 functional. The functional megaspore develop into female gametes or embryo sac. This method of embryo sac formation from a single megaspore is termed as monosporic development. Single megaspore is known as monosporic development. Development of female gamete. The nucleus of the functional megaspore divides meiotically to form 2 nuclei, which move towards the opposite pole, forming a 2-nucleate embryo sac. 2 more sequential mitotic nuclear division result in formation of 4 nucleated and later 8 nucleated stage. These divisions are strictly free nuclear. Nuclear divisions are not followed immediately by cell wall formation. After 8 nucleate stage, the organization of the typical female gametophyte or embryo sac takes place. Generally, 6 of the 8 nuclei are surrounded by cell walls and organized into cells. The remaining 2 nuclei, called polar nuclei, are found below the egg apparatus in the large central cell. Distribution of cells within the embryo sac. The 3 cells containing 2 synergids and 1 egg cell, which are grouped at micropylar end, consist of egg apparatus. The synergids have special cellular thickenings at the micropylar tip called filiform apparatus. The filiform apparatus helps to guide the pollen tubes into the synergids. 3 cells at the chalacil end are known as antipodals. Thus, a typical mature angiosperm embryo sac at maturity is 8 nucleated and 7 cells. 8 nucleated and 7 cells. We saw this in the diagram before, all right? 8 nucleated, 7 cells, where 2 nuclei combine to form polar nuclei, central cell, forming 1 cell. So this is all about megasporangium. All right? Megasporangium is one of the important questions. Make sure you go through the data. All right? Make sure to go through the data. Gynoecium, I'll give you some time. Go through this data immediately. Can we move to the next page, Ma?
Yes, sir.
All right. Can we move to the next page, Ma? Okay.
Sir,
All right, the final stage. After this, we'll be moving into the next topic, second topic. We told there are 3 topics, isn't it? We'll move into the second topic. All right, Ma, we are moving to the next topic. Next topic, pollination and fertilization. All right, pollination and fertilization. This is also very important. We saw that a lot of questions are from this. So, what are the revision notes for this? Modes of pollination. The process of transfer of pollen grains from anther to stigma of pistil is known as pollination. Transfer of pollen grains from anther to stigma is known as pollination. There are few external agents which help in the plants for pollination to take place. Pollination is of 3 types: autogamy, geitonogamy, xenogamy. Right? Autogamy, geitonogamy, and xenogamy. What is autogamy? When the pollen grains are transferred from the anther to the stigma of the same flower, anther to stigma of same flower, known as autogamy. In flowers with exposed anthers and stigma, a complete autogamy is rare, and hence the anther and stigma should lie close to each other to enable self-pollination. Along with this, there should be a synchrony in pollen release and stigma resistivity. We already worked on vitality of pollen grains, isn't it? The maturity of pollen grains and the stigma resistivity, accepting the pollen grains by stigma should be receptive, or else it won't happen. The plants like viola, common pansy, oxalis, and commelina produce two types of flowers, namely chasmogamous flowers and celistogamous flowers. What is chasmogamous flower? They are similar to flowers of other species with exposed anthers and stigma. Chasmogamous, exposed anthers and stigma. What about celistogamous? They do not open at all. Anthers and stigma lie close to each other. They are autogamous, as there is no chance of cross-pollination. When anther dehisces in the flower buds, pollen grains come in contact with the stigma for pollination. Celistogamous flowers produce assured seed set, even in the absence of pollinators. Xenogamy. When the pollen grains are transferred from the anther to stigma of another flower of the same plant, anther to stigma of another flower of the same plant, it is known as Xenogamy. It involves pollination with the help of pollinating agent. It is structural cross-pollination, but genetically self-pollination. It is genetically similar to autogamy because the pollen grains come from the same plant. Xenogamy. When the pollen grains are transferred from anther to stigma of different plant, it is known as xenogamy. Anthers to stigma of different plant, it is known as xenogamy. It brings about genetically different types of pollen grains to the stigma. These are the three types. What are the agents of pollination? There are two types of agents of pollination, namely biotic agent and abiotic agent. What are abiotic agents? There are two abiotic agents, namely wind and water, which help pollination to take place. Pollination by wind pollination taking place by wind is also known as anemophily. Wind and water pollinated flowers are not very colorful. They do not produce nectar. Wind pollinated flowers often have a single ovule in each ovary. Numerous flower remains packed in an inflorescence. Example, corncob, tassel. Tassels are the stigma, style which wave in wind to trap the pollen grains. Wind pollination is commonly seen in grass. Characteristics of anemophilous flowers. The flowers produce enormous amount of pollen. The pollen grains are light, non-sticky, so that they can be transported in wind currents. They often possess well-exposed stamens for easy dispersals of pollen grains. They have large, feathery, and sticky stigma to trap the airborne pollen grains. Pollination by water. Pollination takes place in water. The pollination taking place in water is called hydrophily. It is limited to about 30 genera, mostly monocotyledons. In Vallisneria, the female flowers reach the surface of the flower by a long stalk, and the male flowers or pollen grains are released on the surface of water. These male flowers or pollen grains are carried by water currents and reach the female flowers. In sea grasses, the female flower remains submerged in water and the long ribbon-like pollen grains are carried inside the water and reach the stigma. The pollen grains of most water pollinated species have mucilaginous covering to protect from wetting. Not all aquatic plants use hydrophiles. For example, in aquatic plants like water hyacinth, water lily, etc., the flowers emerge above the level of water for entophilic or anaphilic, for pollination to take place by insect or wind. It is seen in Wellisneria and Hydrelia, freshwater, Zostera, marine seagrass, etc. Water pollinated flowers. Okay, water pollination by water. Okay. What are the biotic agents? What are the biotic agents? Some flowering plants use animals as pollinating agents like bees, butterflies, flies, beetles, wasps, ants, moths, birds, sunbirds, and hummingbirds. Bats, some primates, lemurs, arboreal tree-dwelling rodents, reptiles, gecko lizards, and garden lizards. Bees, butterflies, flies, beetles, wasps, ants, moths, birds, bats, primates, some primates, arboreal tree-dwelling rodents, reptiles, etc. When the pollination takes place by insect, it is known as entomophily. Often, flowers of animal pollinated plants are specifically adapted to particular species of animal. When the animal comes in contact with anthers, body gets coating of pollen grains. When it comes in contact with stigma, it results in pollination. Some plants provide safe places as floral reward to lay eggs. Example, Amorphophilus, the tallest flower. It will provide place for laying eggs, safe places, because eggs can be easily eaten by predators. There is a very close obligatory symbiotic relationship between species of moth, Pronumba, and the plant Yucca. They cannot complete their life cycle without each other. The moth deposits in eggs in the locule of the ovary, and the flower gets pollinated by the moth. The larva of the moth come out of the egg as the seed starts developing. See here, there is a symbiotic relationship between the species and this moth. They cannot complete their life cycle without each other. Moth deposits its eggs in the locule of the ovary, and the flower gets pollinated by the moth. The larva of the moth come out of the egg as the seed starts developing. Many insects consume pollen or nectar without bringing about pollination. They are called pollen or nectar robbers. The characteristics of entomophilous flowers. The flowers are large, colorful, fragrant, and rich in nectar. When the flowers are small, they form inflorescence to make them visible. Example, sunflowers. The sunflower, each and every small petal we think, the yellow petal-like thing, it is, each and everything is a male, right? The flowers pollinated by flies and beetles secrete foul odor to attract these animals. The pollen grains are generally sticky. Next, this is about the agents of pollination. Outbreeding devices, device for promoting cross-pollination. This is also a very, 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 very important topic. To avoid self-pollination, cross-pollination is encouraged in plants as follows.
avoiding synchronization. In some species, pollen release and stigma receptivity are not synchronized. Either the pollen is released before the stigma becomes receptive, or stigma becomes receptive before the release of pollen. The anther and stigma mature at different times. This phenomena is known as dichogamy. It prevents autogamy, that is, pollination in same flower. Arrangement of anthers and stigma at different positions. In some species, the arrangement of anthers and stigma at different positions prevents autogamy. Self-incompatibility. It is a genetic mechanism that prevents pollen of one flower to germinate on the stigma of the same flower. Pollen of one flower germinate on the same flower because different pollen grains comes and get settled in the stigma, but only the pollen of the same flower can germinate, or else it won't be germinating. Production of unisexual flowers. Monoecious plants such as castor, maize, where the male and female flowers are present on the same plant, prevents autogamy but not gametogamy. On the other hand, dioecious plants like papaya, where male and female flowers are present on different plants, prevents both autogamy and gametogamy. What is dioecious plant? Dioecious plant. Different male flower would be in different plant, another plant of papaya, male plant, and female would be in another plant. That is dioecious. Monoecious, same plant would be having both male and female flowers. Bisexual flowers, same flower would be having both male and female. Okay. Why only the same kind of pollen is getting pollinated? That is go through fertilization due to the pollen tissue interaction. It is a dynamic process involving pollen recognition followed by promotion or of the pollen. Right? A stigma recognize this pollen is from my genes. Automatically, it will promote the pollen tube to enter it, or else from the different one, it will inhibit the pollen. This interaction takes place through chemical component produced by them. If the pollen is compatible, then the pistil accepts it and promotes post-pollination event. The pollen grain germinates on the stigma to produce pollen tube through germ spore, which is an exine, the only place which doesn't have sporopollen. The content of pollen grain move into pollen tube. The pollen tube grows through the tissue of the stigma and style and reaches the ovary. If the pollen is incompatible, the pistil rejects the pollen by preventing pollen germination on the stigma or the pollen tube growth in this time. In some plants, the pollen grains are shed by two-celled stage. The generative cell divides and forms two male gametes during the growth of the pollen tube and stigma. In plants that shed pollen in three-celled stage, the pollen tube carried two male gametes from the beginning. The pollen tube, after reaching the ovary, enters the ovule through the micropyle, chalaza and integuments, and then enter one of the synergids through the filiform apparatus. The filiform apparatus present on the micropylar part of synergids guides the entry of pollen. The plant breeder can manipulate pollen pistil interaction even in incompatible pollination to get desired hybrids. So this results in artificial hybridization. What is this artificial hybridization? It is one of the major approaches of crop improvement program by using desired pollen grains for pollination. This is achieved by emasculation and bagging techniques. Emasculation is the removal of anthers by using forceps from the bisexual flowers before the anther dehisces. The emasculated flower are then covered with suitable bag made up of butter paper to prevent contamination on its stigma with unwanted pollen. This is known as bagging. When the stigma attains receptivity, the mature pollen grains collected from anthers of male parent are dusted on the stigma. Then the flower are rebagged and allowed to develop the fruit. If the female parent produces unisexual flowers, there is no need for emasculation. The female flower buds are bagged before the flower opens. When the stigma becomes receptive, pollination is carried out using the desired pollen and the flower rebagged. Good. This is known as artificial hybridization. This is known as artificial hybridization. Then, this is also one important 3 marks and 5 marks also they ask about this artificial hybridization. Then, the last part of the second topic. The last part of the second topic is double fertilization. The pollen tube, after entering one of the synergy, releases its contents, including 2 male gametes, into the cytoplasm of the synergids. One male gamete moves towards the egg cell and fuses with the nucleus by the process of Xenogamy. We already know what is Xenogamy. The process of combination of male gamete and female gamete to form diploid condition. 2 haploid one male gamete and female gamete to form a zygote is known as Xenogamy. To form diploid cells, zygote. The other male gamete moves towards the 2 polar cells located at the central cell, fuses with them to produce triploid primary endosperm. Triploid primary endosperm nucleus. 10. As this involves the fusion of 3 haploid nuclei, it is called triple fusion. Since 2 types of fusion where Xenogamy and triple fusion take place in the embryo sac, this is known as double fertilization. The central cell after triple fusion becomes the primary endosperm cell. Primary endosperm cell, PEC. Primary endosperm cell, PEC, develops into endosperm, while the zygote develops into embryo. The tender coconut we drink, the liquid we drink, is made up of primary endosperm cells, which nourishes the cell to form that white thick coconut we are using for kitchen. It is an event unique to flowering plants. It is an event unique to flowering plants alone, right? Very good. With this as a base, we complete the second topic. We have one small topic. Which would be taking only another 20 minutes or so. Only another 20 minutes or so. After 5 minutes of break, we will start with that session and complete the whole lesson. Right? Okay. Well and good. Thank you, Doctor. Sister, did you understand it? Sister, did you understand it?
Yes, sir.
All right. Very well done. Very well done. Okay. Good. We will, after 5 minutes, we will see through the next topic and complete the whole lesson. Then I will send you the, question paper for both the lessons. Take your leisure time and complete that topic. Right? Okay,